Hello everyone, and welcome to my presentation of Saccharides, carbohydrate of physiological significance for our Chem 1050 biochemistry course. My name is Håknar Spalnod, and I'm excited to discuss why carbohydrates are so crucial for life. Today we will explore what saccharides are and why they are vital for us. We will look at their classifications, monosaccharides, disaccharides, and polysaccharides, and how they affect our health. We will also discuss how they related to conditions like diabetes and their importance in clinical care. On this slide, we are defining saccharides as polar molecules primarily made of carbon, hydrogen, and oxygen. Their power mainly lies in three key areas. First and foremost, energy metabolism. Glucose is the best known carbohydrate fuel, entering pathways like glycolysis and citric acid cycle to produce ATP. Second, structural integrity. In plants, cellulose forms rigid cell walls. In animals, glycogen serves as a rapid metabolizable energy reserve. Third, cell signaling. Carbohydrate chains on glycoprotein and glycolipids help cells communicate and recognize pathogens and mount immune responses. By grasping these fundamental roles, we will better appreciate how carbohydrates underpin critical metabolic and physiological processes throughout this presentation. Learning objects. There's a few learning objects, and I want to start by outlining them. By the end of this presentation, you should be able to first classifying carbohydrates into the three major groups: monosaccharides, disaccharides, and polysaccharides. And second, explaining the different types of carbohydrate isomers. Third, identify how these molecules function in energy metabolism, providing fuel for our cells as well as in structural roles. Fourth, evaluate the glycemic index, understanding how different carbohydrates affect our blood glucose levels and why this matters for metabolic health. And last, recognize the clinical implications of saccharides, especially in conditions like diabetes, where carbohydrate metabolism becomes impaired. Keep these points in mind as we move forward. Types of saccharides. Carbohydrates or saccharides can be broken into three main categories. First, monosaccharides. These are the simplest form of carbohydrates. Common examples are glucose, fructose, and galactose. They typically have 3 to 7 carbon atoms, but the most biologically significant ones are 5 or 6 carbon sugars. Glucose, or for instance, is our body's primary energy currency. It circulates in the bloodstream and is immediately available for ATP production. Second one is disaccharides. These consist of two monosaccharides joined together by a glycemic bond. Examples include sucrose, lactose, and maltose. Disaccharides often need to be broken down into their monosaccharide components before our body can absorb them. Third, polysaccharides. These are large complex carbohydrates composed of multiple monosaccharides. Common polysaccharides include glycogen, the main storage form of in animals, stored in muscles and liver; starch, energy stored in plants; and cellulose, the primary structural component in plant cell walls. Polysaccharides can have diverse structures and functions, from providing quick energy reserves to contributing to rigid plant fibers. On this slide, we're going to talk about structural representations. Monosaccharides like glucose can be drawn in several ways, each providing different insights into their chemical properties. First one is a linear form. This shows glucose as a straight chain aldohexose. It's useful for illustrating oxidation and reduction reactions, and is how the molecule often appears in non-aqueous or theoretical contexts. And the second one is Haworth projection. Once glucose cyclizes, um, the Hilbert projection provides a simplified ring depiction. This lets us see the alpha or beta orientation of the hydroxyl group at the anomeric carbon, an important factor in glycosidic bond formation. The third one is chair form. The chair conformation is the most realistic 3D arrangement of the six-membered ring, minimizing steric strain. It helps us visualize which groups are axial and which groups are um, equatorial, both of which affect the sugar's stability and interaction with enzymes. And forms of isomerism. Carbohydrate exhibits several fascinating forms of isomerism which can drastically alter their biochemical behavior. The first one is stereoisomerism, D and L forms. These forms related to the orientation of the hydroxyl group around the chiral carbons. The D and L designations are based on the configuration of the chiral center furthest from the carbonyl group. In nature, these sugars are overwhelmingly more common in human biochemistry than L sugars. The second one is anemorism, alpha and beta. When a linear monosaccharide cyclizes, the new ring introduces an anaerobic carbon, which can have the substituent hydroxyl either or on the same side as the ring substitutes or on the opposite side, alpha. This difference is crucial for glycosidic bond formation. For instance, starch and cellulose are both polymers of glucose, but the orientation of the glycosidic bonds, alpha versus beta, leads to dramatically different properties. Third one is we're going to talk about apimerism. This is the different at just one chiral carbon. For example, glucose and galactose differ at the C4 position, making them apimers. This tiny structural variation means different enzymes handle these sugars differently. And next one is the aldose and ketones isomerism. Sugars can also differ by having either an aldehyde group or a ketone group. Glucose is an aldose, fructose is a ketose. Yet both share the same molecular formula. This difference dictates how they behave in metabolic pathways. Fructose is metabolized slightly different than glucose, especially in the liver. Biological functions. Let's explore these three major biological functions more closely. First, energy production. Carbohydrates, particularly the glucose, are central to ATP production. In glycolysis, glucose is broken down into pyruvate, yielding a modest amount of ATP, and then pyruvate enters further pathways like the citric acid cycle and oxidative phosphorylation, where more substantial ATP is generated. This stepwise process is efficient and tightly regulated to meet the energy demand of cells. And the second role is structural roles. In plants, cellulose fibers form rigid cell walls, supporting growth against gravity. Humans cannot digest cellulose due to lacking the enzyme cellulase, but it remains an important source of dietary fiber. In animals, glycogen is stored primarily in the liver and muscles. It's highly branched, allowing enzymes to release glucose rapidly when blood sugar drops or during intense exercise. Third, and signaling. Many glycoproteins and glycolipids line the surface of cells. These Carbohydrates tags help cells recognize one another, crucial for immune function, hormone targeting, and even fertilization. Defects in these structures can lead to immune disorders or issues in cellular communication. Um, Glycemic index and metabolic impact. The glycemic index is a valuable tool for understanding how fast and how high a particular carbohydrate containing food raises blood glucose level after consumption. Here's why it matters. First, high GI foods. Examples include glucose itself, white bread, white rice, or sugary beverages. These lead to a rapid spike in blood sugar, triggering a corresponding spike in insulin release. Over time, frequent blood glucose peaks may contribute to insulin resistance, weight gain, and other metabolic issues. Low glycemic index foods, think whole grains, legumes, or certain fruits and vegetables, these release glucose more gradually, helping maintain more stable blood sugar levels. This often correlates with better long-term metabolic health. Clinical relevance is people with diabetes or pre-diabetic needs to monitor their carbohydrate intake carefully, not just in terms of quantity but also quality. Choosing lower glycemic index options can help in controlling postprandial, the after meal glucose levels, and managing insulin requirements. Even in non diabetic populations, focusing on moderate to lower glycemic index meals can support weight management and reduce the risk of developing metabolic syndromes. Thus, the glycemic index is an important concept in both nutrition science and public health. Understanding how different carbohydrates behave in the body enables better dietary planning and preventive strategies against chronic conditions like diabetes and obesity. Um, this slide is about conclusion. To summarize what we learned today, carbohydrates or saccharides are fundamental molecules in biology, fueling our cells, building structure components, and assisting in signaling. From simple monosaccharides like glucose to long polysaccharides like starch and glycogen, their versatility is immense. We have seen how isomerism can alter carbohydrate function and how the glycemic index help us gauge their metabolic impact. Clinically, this knowledge is crucial for managing diabetes, obesity, and other metabolic conditions. By making informed dietary choices and understanding carbohydrate metabolism, patients and healthcare providers can better control blood glucose, maintain energy balance, and reduce complications associated with metabolic disorders. I hope this overview clarifies why carbohydrates really are at the heart of biochemistry and human health. And before wrapping up, let's do a quick review with these multiple choice questions. First, which of the following is the disaccharide? Glucose, fructose, or sucrose, glycogen? The answer is fructose. Second one is what type of bond joins two monosaccharides? That would be glycosidic bond. And third one would be which term describes two sugar molecules that differ only in the orientation of the hydroxyl group at their anomeric carbon? The answer is B, anomers. For primary storage form of carbohydrate in animals is glycogen. The last one is which carbohydrate has the highest glycemic index? is glucose or sucrose. It depends on what you look at. Usually glucose. Thank you so much for your attention throughout this presentation. I hope you found it informative and that it's given you a clearer sense of how carbohydrates function and why they are so crucial to life and health. If you have any questions, please let me know. Thank you very much.